Welcome back to the deep dive. Today we are taking on, well, an incredible journey into our own beginning.
Human embryology.
Exactly. And we're focusing on the very, very foundations, the first couple of weeks, what's known as preimplantation development.
This is the period that, it really sets the stage for everything that comes after.
So to navigate this, where everything is happening so fast on a microscopic level, what's our guide?
Our guide is the gold standard,
Oh.
the Carnegie system of embryonic staging. And what's really crucial for you to get here is that these stages, all 23 of them, are based purely on morphology.
Morphology,
Yeah.
so shape and structure.
Exactly. It's about what the embryo looks like, its internal and external complexity, not how old we think it is in days.
And that distinction is so important. Our sources point out that the timing can be really unreliable. For stages 6 through 16, they think that embryos might be 3 to 5 days older than we used to estimate.
Right, so the stage is the universal language. You should think of it less like a timeline and more like a checklist of architectural achievements. If the structure is there, the stage is met.
Regardless of whether it took 4 days or 7 days to build it.
Precisely. And our mission today begins at stage 1, fertilization.
The very beginning. And it happens fast, usually high up in the uterine tube in the ampullary region.
Typically within 24 hours of ovulation.
But the sperm that gets there first isn't actually ready to go, is it? It has to go through this process called capacitation.
Yeah, that's right. It's a series of membrane modifications, almost like a final activation sequence that prepares it to interact with the egg.
Once that's done, it still has to fight its way through these outer layers, the cumulus oophrus and corona radiata.
To get to the main barrier, this thick glycoprotein shell called the zona pellucida, and binding to a specific receptor there, ZP3, is the signal.
The trigger.
The trigger for the acrosome reaction, which is this dramatic, irreversible event.
I always picture it like a microscopic excavation team. The sperm releases these powerful enzymes like a crucin.
And they just digest a tunnel.
A path right through the zona pellucida so it can finally make contact with the oocyte membrane.
And that contact, that fusion, is the cue for maybe the most vital safety feature in all of early biology. It triggers an instantaneous spreading calcium wave.
A calcium wave.
Imagine a ripple spreading across a pond. This wave crosses the entire egg in like 5 to 20 seconds. It's incredibly fast.
And that wave is not just the master switch for development, it's also the ultimate safety lock. It establishes the block to polyspermy.
Exactly right. That surging calcium causes these little sacs called cortical granules to fuse with the egg surface.
And they release what?
Enzymes. They release enzymes that flood the space right under the zona pellucida, and they basically chew up the ZP3 receptors.
So no other sperm can bind.
It immediately makes the zona impenetrable. It's a chemical shield that ensures only one set of paternal DNA gets in.
Okay, so the genetic material is safely inside. Now, the source material points out this really subtle but fascinating detail about the pronuclei.
Oh, this is a great point.
The maternal and paternal sets of chromosomes, they form their own little nuclei, but they don't actually fuse into a single nucleus.
Yeah, that's a huge conceptual twist, isn't it?
Yeah.
We think of a zygote as a single cell with one fused nucleus. But here, the nuclear envelopes just disappear. And the chromosomes line up directly on the first cleavage spindle.
So technically, the first true diploid nucleus isn't formed until the two-cell stage?
Correct. It's an incredible detail.
That's just astonishing. And how does this play into the next big concept, parental imprinting?
This is where we see this functional inequivalence, a kind of genetic negotiation between the parents.
What do you mean by negotiation?
Well, the paternal genome is disproportionately essential for building the extraembryonic tissues.
So the placenta.
Right, the placenta. On the other hand, the maternal genome is relatively more important for developing the embryo itself.
It almost sounds like the placenta and the baby have different genetic owners. Why would evolution set it up like that?
The theory is it's an evolutionary balancing act, often related to growth. Paternally expressed genes tend to push for more growth, while maternal genes tend to suppress it.
And we see what happens when that goes wrong.
We do. In disorders involving the 15q11.13 chromosome domain, if the paternal copy is incorrectly imprinted, you can see Prader-Willi syndrome, a growth disorder.
And if it's the maternal copy...
Then you can see Angelman syndrome, which is a neurological disorder. The whole fate of the organism can hinge on which parent's gene is switched on.
Amazing. Okay, so moving into stage 2, we get these cleavage divisions. The cell number just skyrockets. 2 cells, 4, 8.
But the embryo itself isn't getting any bigger. The cytoplasm is just being divided among smaller and smaller cells, the blastomeres.
And who's in charge at this point?
The mother. Entirely. Up to the 8-cell stage, the embryo is running on the mRNA and proteins she packed into the egg.
And the embryo's own genes don't kick in until...
Not until around 55 hours, right at that 8-cell checkpoint. That's when its own mRNA transcription starts.
And once we hit that checkpoint, everything physically changes. We get this process called compaction.
Yes, and this is a huge transformation. Instead of a loose clump of spherical cells, they suddenly flatten against each other. They maximize their contact.
It's like a bunch of loose marbles suddenly snapping together into a tightly packed cube.
That's a great way to put it. This relies heavily on an adhesion glycoprotein, E-cadherin, that basically glues them together.
And this very process, compaction, it starts to decide the cell's fates. Lineage commitment.
It does. It all comes down to how they divide. If the division is perpendicular to the cell's polarity, you get two different daughter cells.
Okay.
One is a polar cell on the outside, and one is a polar cell deep on the inside.
So position is destiny, even at this stage. The outer polar cells, they become what?
They'll form the trophectoderm, which is the future placenta, while the deeper apolar cells form the inner cell mass, or ICM.
Which is the future baby.
Exactly. And that first critical decision is made by the 16-cell stage, the morula.
This then drives the next step, blastocyst formation, stage 3. This is around day 4.
Right. The outer trophectoderm cells finish their job. They form tight junctions, creating a sealed container.
And once it's sealed, you said it needs to inflate.
It does. The outer cells start acting like tiny water pumps. They pull fluid from the environment into the center, generating this fluid-filled cavity, the blastocyst.
And the final step before it can implant is hatching. It has to escape the zona pellucida.
Yes, around day 6 or 7. And the visualization is pretty remarkable. The embryo often squeezes out in a sort of figure-of-8 shape.
And the source material mentions this could be related to twinning.
It's one potential mechanism, yeah. If that emerging mass of cells gets pinched and separates into two distinct entities, you could get monozygotic twins.
So now the blastocyst is free. By stage 5, the inner cell mass organizes itself into the bilaminar embryonic disc.
And this is so fundamental. You get two clear layers. The upper layer, closest to the uterine wall, is the epiblast. The lower layer is the hypoblast.
And just by creating these two layers, it establishes the top and bottom, the dorsiventral axis.
That's it. The fundamental body axis is set right then and there.
Okay, so the implantation process itself, 7 to 12 days. It's invasive.
Highly invasive. The blastocyst has to adhere to and then literally invade the maternal endometrium.
Right.
And the tissue doing the invading, the syncytia trophoblast, is incredibly aggressive.
Which has immediate clinical importance.
Absolutely, because that invading tissue starts pumping out human chorionic gonadotrophin, HCG.
The pregnancy hormone?
The very one. And it's detectable in maternal blood as early as 6 to 8 days post-fertilization. It's our earliest reliable sign of pregnancy.
Let's talk about the clinical side of that invasion. The big danger here is ectopic implantation.
Right. When the blastocyst implants somewhere, it shouldn't, most often in the uterine tube.
And what makes that so dangerous?
The syncytia trophoblast doesn't care what it's invading. It will burrow into any tissue it touches, and the wall of the fallopian tube is fragile.
So it can erode blood vessels?
And cause a catastrophic hemorrhage. It's a true medical emergency.
Speaking of interventions, let's touch on Assisted Reproductive Technologies, RT-IVF, ICSI.
Yes, especially intracytoplasmic sperm injection, ICSI, where a single sperm is injected directly into the oocyte. It's been revolutionary for male factor infertility.
But the source material flags a warning. It says ARCI is associated with an increased risk of some of these imprinting syndromes we mentioned, and also ectopic implantation.
It is. This is a critical area of ongoing research.
The source mentions that RT challenges the temporal conversation between the embryo and the mother. What does that mean?
It means that by taking the embryo out of the uterine environment for those first few crucial days of culture.
During compaction and blastocyst formation.
Exactly. We might be disrupting the environmental signals it needs to correctly set its epigenetic markers, like the ones that control parental imprinting. The risk is small, numerically, but the consequences of these syndromes are severe.
Okay, let's look at the other structures the embryo is building to support itself. The amniotic cavity.
This actually forms within the epiblast cells. A space opens up, fills with fluid, and creates that protective, fluid-filled sac for the future embryo.
And below that, the hypoblast forms the yolk sac.
That's right. The hypoblast cells spread out. creating a boundary for the primary yolk sac, which is the embryo's first source of nutrients.
But the hypoblast has another, even more fascinating role, a signaling role.
Yes, this is incredible. The visceral hypoblast cells, ones right next to the epiblast, they are crucial for telling the epiblast where to form the primitive streak.
So they're basically pointing to the midline and saying the main body axis goes here.
Precisely. Without that signal, the embryo can't organize itself.
And then we have the extra embryonic mesoblast providing the structural support,
Right? Fills in the space, and eventually it gets restricted down to form the connecting stalk.
which becomes the umbilical cord.
It becomes the foundation for the vascular pathway in the umbilical cord, yes. The lifeline.
Okay, a final interesting divergence, twinning. We have dizygotic, or fraternal, twins from 2 eggs.
And monozygotic, identical twins.
Hmm.
From one egg that splits.
And the risk all depends on when that split happens.
It does. And clinically, it comes down to the membranes they share. The most dangerous situation is a monoamniotic, monochorionic placenta.
Meaning they share both the inner sac and the outer sac.
Yes. And that carries about a 50% perinatal mortality rate.
50%. Why so high?
Yeah, because of complications like their umbilical cords getting tangled or dangerous vascular shunts forming between them in the shared placenta.
Wow. That really puts into perspective how critical these early structures are. Let's just quickly recap the sequence.
Okay. We started with the Carnegie system, prioritizing morphology over age. Then the incredible speed of fertilization, with that calcium wave as a protective lock.
Then we walk through cleavage and compaction, where E-cadherin glues the cells together and sets up that first big decision. Future baby or future placenta.
We tracked the aggressive invasion of the syncytiotrophoblasts during implantation and the serious danger of ectopic pregnancies.
And finally, the supporting cast. The amnion, the yolk sac, and especially the hypoblast, acting as this hidden director that establishes the entire body plan.
Which brings us back to that question about art. Since it involves that early in vitro culture, challenging that exchange of environmental information, what are the broader implications?
So what does this all mean? Consider that these incredibly complex processes, like setting those epigenetic marks for parental imprinting, they aren't just driven by a fixed genetic code. They are influenced by environmental conversations that start days before implantation.
We are literally being shaped by invisible, instantaneous biological cues, all happening within a span of about 14 days.
So the question to leave you with is, what other hidden early conversations are shaping us that we have yet to fully understand?
A fascinating thought.
Thank you for joining us for this deep dive into the earliest, most complex days of human development. We really hope this knowledge sticks with you.